Macromolecules in your book, and it's the activity C of the hydrolysis of carbohydrates. First of all, you'll have 8 tubes that you'll label 1 through 8. Then you'll have 2 big tubes, okay? One will be labeled sucrose and the other one will be labeled starch, okay? Now, what you'll do is you'll put 10 milliliters of sucrose solution that will be labeled in your lab, and you'll put it into one tube that says sucrose, okay? You do the same thing with the starch. You'll put 10 milliliters of starch into a big tube that's labeled starch from your lab, okay? Now, from there, you'll actually start a water bath. You'll need this water bath for several activities, okay? Once you've got your water bath going and you've got your 2 big tubes of starch and of sucrose, you're going to add 5 mils to each tube, okay, of hydrogen chloride, okay, so your HCl, you're going to add, so you should have 10 mils of sucrose and 10 mils of starch, and to each tube you will add 5 milliliters of HCl, okay. You'll mix those gently, both of those, okay. Now, with your 8 tubes, in tube 1, 3, and in tube 4, okay, in tube 1, you'll actually take 1 pipette full, okay, you take 1 pipette full of your sucrose solution and you'll add it into tube 1, okay. Then you'll take a different pasture pipette, okay, and in tube 3 you actually add your starch. So you'll take the other one and you'll do starch, 1 pipette full into tube 3, okay, and same thing with tube 4, okay. Okay. Now, after you've done that, you'll place your big tubes, both of these, in your boiling water bath, okay? Then after that, okay, let's find this one. After 2 or 3 minutes, you'll take sucrose from your big tube that you've got in your boiling water bath, and you'll place 1 pipette full into tube 2, the one that we skipped, okay? Now you're done with the sucrose, okay? You can put that to the side. So now we're going to work on the starch. After 5 minutes of your starch has been in the boiling water bath, you're going to take 1 pipette full and put it into tube 5, okay? Then another tube, you're going to take the same starch and do tube 6 with, okay? Now you're going to wait for about 10 seconds. Actually, hold on. You're going to wait 10 more minutes, okay? And then you're going to repeat the one for tube 6 and for tube 5, okay? And you're going to put that into tube 7 and 8, okay? So, once you've got 5 and 6 done, you'll wait for about 10 minutes, then you'll take this tube and put it into tube 7 and 8. Oh, gosh. Now, what you'll do is tubes 1, 2, 3, 5, and 7, okay? So 1, 2, 3, 5, and 7. You'll place those in the boiling bath for 5 minutes, okay? And to that, you're going to add a few drops of Benedict's reagent, which should be a blue reagent, okay? It will be labeled Benedict's. Make sure it says Benedict's on there, okay? Very important. Once you put the dropperfuls into these tubes here, then you'll put them in boiling water bath for the 5 minutes, okay? Now to tubes 4, 6, and 8, what you're going to do is add 2 or 3 drops of iodine, okay? So that's this little yellow solution we always know about, okay? A couple of drops of those, okay, into there, okay? And then what you do is swirl it around and record the results, the color results on your next table, on table 1, okay? Then after you've had the water bath, for those that were sitting in water bath for 5 minutes, you'll let them cool for a second, and then you record the color results in your table once again.